Okay, we're going to talk about Krebs cycle, which is the center of metabolism for humans. And this first slide I'm showing you, right now it'll seem a little complex, but I guarantee you this is a really key thing that you'll be glad you know once we get past it. And I'm just going to go over the highlights. And the reason I'm going over Krebs cycle is it's a key thing to have a general understanding of to understand cancer. It's also a key thing to understand for just general metabolism. We don't need to know all the details, but there's a few general ideas that'll be real helpful to you, and they apply to tons of things. Origin of life chemistry, just health and nutrition in general. All right, so this diagram is the inner mitochondrial membrane, and I'm going to show the slide again later in the talk, but just so you have a general idea of it now, it'll help you. So what happens with glycolysis, Krebs cycle, is that electrons are torn off of glucose. And the reason you take off the electrons is you can use them to make energy. A hydrogen proton, so this is 24 hydrogen protons, are ripped off of glucose. And then they're split. They're split into electrons, 24 electrons, and 24 protons. H plus is a proton. It's really just, a proton is like the hydrogen nucleus. An electron, a typical hydrogen, has one proton and one electron. That's what hydrogen is. All right, so the electrons are passed through the electron transport chain in the inner mitochondrial membrane, and they go from the first complex one, CX1, and then they're passed progressively to more aggressive electron grabbers. And then they get to complex 3, and finally to complex 4, and then the electrons are passed to oxygen. Oxygen is the ultimate electron acceptor. It has a very high electronegativity, meaning desire to grab electrons. And then the oxygen also is combined with hydrogens and it makes water, H2O. And so this is the opposite of photosynthesis. So photosynthesis is to make glucose. And then human metabolism is to break glucose down into water. Okay, and carbon dioxide. So the key point about electron transport is as you move from the first one all the way down to the last one, the desire to grab those electrons gets stronger and stronger. So it's like rolling a ball downhill to the most stable energy situation is when the electrons are given to oxygen. And this was what it's all about. This is why we breathe oxygen. This is why we need oxygen to exercise and for energy, is because this oxygen as the ultimate electron acceptor is what enables the entire electron transport chain to run. And then the electron transport chain is coupled to the pumping of protons. So a proton is pumped out at complex 1. A proton, the H plus here, is pumped out at complex 2. Complex 3. Complex 2 actually doesn't pump any protons. Complex 4 also pumps a proton out. Now you'll have built up a gradient of protons, and this space is IMS for intramembranous space. And then those protons are going to be sucked back down through this structure here. This protein is called ATP synthase, and that energy obtained by harvesting the gradient, if you will, letting the proton come back into the mitochondrial matrix. This is mitochondrial matrix, is used to make ATP, to convert ADP into ATP. And this is the secret of energy for life on Earth, for all multicellular organisms. This is it right here. I realize that's a lot of complexity to throw at you for the first slide, but this general idea, we'll explain it in more detail. All of life revolves around this. And cancer messes this up. So that's going to be real valuable for understanding cancer. And the reason why you want to understand cancer is it'll help you to avoid it, etc. Okay, so here's just the anatomy of a mitochondria. These are all in order here. OMM is outer mitochondrial membrane. So here's the outer mitochondrial membrane right here. And then here is the intramembranous space. So the intramembranous space is located at the tip of this red dot here between the outer mitochondrial membrane and the inner mitochondrial membrane. This is where those protons are being pumped into this intramembranous space. It's like proton exile. All right. And now here's the IMM, intra-mitochondrial membrane, inner mitochondrial membrane. So that's the one that's where all those proton pumping complexes are located on this inner mitochondrial membrane. And it's kind of convoluted to increase surface area. Any given individual cell can have 1,000, 2,000 mitochondria. Some have even more than that. So they make a lot of ATP. Okay, here is a mitochondrial matrix. A mitochondrial matrix is just the center. It's all colored yellow on this slide inside the matrix. So this is where Krebs cycle runs, inside the mitochondrial matrix. and then electron transport and oxidative phosphorylation occur on this inner mitochondrial membrane. Okay, so we just talked about it. Here's a hydrogen, is just H, and a hydrogen has one proton and one electron. So when you remove the electron, all you're left with is a proton. And so what I'm saying is about 24 of these are yanked off of glucose, and then the electrons are passed down that electron transport chain, whereas the protons are pumped out. Okay, and the way I'm explaining this is a little bit of an oversimplification, but it's what you need to know. And so the key thing here is just to get the couple key points, because they're going to be the foundation of understanding tons of other things. Okay. All right, so now here is Krebs cycle. Krebs cycle. Kreb is the name of the biochemist scientist Hans Krebs who discovered Krebs cycle, and he was a student of Otto Warburg. So Otto Warburg is the guy who won the Nobel Prize. He discovered the Warburg effect, that hypoxia causes cancer, and then his student Kreb also won a Nobel Prize, and he figured out Krebs cycle. And the incredible thing about Krebs cycle is in cancer, it goes backwards. And the fact that it goes backwards is actually really interesting. And I'll explain why in just one sec. Let's just talk a little bit, though, about how Krebs cycle works. So glucose comes into the cell. It undergoes a metabolic pathway called glycolysis that starts to break it down into a 3-carbon molecule called pyruvate. Whenever you see ATE on the name of a molecule, that means it's a carboxylic acid that's deprotonated. And you're going to see carboxylic acids are super common in human metabolism. Every single molecule in Krebs cycle is a carboxylic acid, okay? So once glucose is broken down to pyruvate, it undergoes a decarboxylation. Decarboxylation means to remove CO2. So the CO2 comes off of it, all right? And now it'll become a 2-carbon molecule like acetate. It's bound to something, a carrier called coenzyme A. We'll just call it CoA. During this step, in addition to taking the CO2 off, you also take off 2 hydrogens. And like I said, these hydrogens will then split into their proton and their electron. Their electron will run down the electron transport chain to generate energy, like water flowing down a hill. Protons will be pumped out with that energy. And then later on, when a proton is brought back into the mitochondrial matrix, that is harvesting the energy of the gradient to produce ATP. And this is how all life on Earth makes energy. This is the foundation of all life on Earth, aerobic energy production. Okay, well anyways, once pyruvate is decarboxylated, the CO2 comes off, it becomes acetyl-CoA. And acetyl-CoA is sort of the central molecule in all of human metabolism. Fatty acids are also chopped up into 2-carbon units called acetyl-CoA. Amino acids, some of them can be converted into acetyl-CoA as well. So acetyl-CoA, then, the pyruvate, this will happen inside the mitochondrial matrix, whereby acetyl-CoA is a 2-carbon molecule, that's what the C2 stands for here, is going to combine with a 4-carbon molecule, that's what C4 stands for, and it's going to produce a 6-carbon molecule. So that 6-carbon molecule is called citrate. So Krebs cycle can also be called the citrate cycle or the citric acid cycle. It's going to have 3 carboxylic acids on it, so it's also called the tricarboxylic acid cycle. And as it goes around in a circle, it will first of all chop off a CO2, then chop off another CO2 here. So it'll lose 2 CO2s, which means 2 carbons. Thus you end up with a C4, add another acetyl-CoA in, and the cycle can run again. But the main thing the cycle does, and the main purpose of the cycle is it's harvesting hydrogens, pulling these hydrogens off of the glucose derivatives. And the reason it wants to pull those hydrogens off so they can be sent to electron transport. Think of a hydrogen as being, think of, then the hydrogen splits into the electrons. It's especially the electrons. Think of those as being energy. It's a way of carrying energy around. Just remember that. That's going to be useful. Okay, so just a couple points. Stealing an electron is stealing energy. Anaerobic metabolism is good enough for a lot of single cell life like bacteria. But essentially all multicellular life runs on aerobic metabolism. Yeah, there's some potential rare exception in deep water, anoxic water with a lot of salt in it. But in general, it's a reasonable thing to consider that basically all multicellular organisms use aerobic metabolism. In general, that's true. Minor exceptions really don't matter for our concern. Okay, aerobic metabolism provides much more energy than you can get with anaerobic metabolism. It's at least 16 times as much energy. Okay, so if you're going to be part of a multicellular organism and have a highly specialized job, like let's say something is a liver cell. A liver cell has to detoxify all kinds of things in the human body, including estrogen. It has to make bile for lipid digestion. It also has to maintain blood glucose levels. So it has to store glycogen. It has to know when to break down glycogen. It has to know when to run gluconeogenesis to generate glucose for the blood. So a liver cell needs tons of energy. It has a big job. It's the metabolic workhorse of the human body. So the point I'm saying is it needs oxygen so it can make all the ATP, make all the energy. ATP is like energy money, like a $20 bill is to humans with cash. All right, so if it doesn't get oxygen, a liver cell can't do its job. It can't make all those things, the bile, etc. So it'll probably die or it'll transform itself into becoming cancer. And that's why hypoxia can cause cancer. That's Warburg effect. Getting back to Krebs cycle. Krebs cycle normally, as far as we're concerned, it runs forward like we just showed it, and it's harvesting electrons. It's like one of those turning combines, and it's harvesting electrons from the derivatives of glucose. So Krebs cycle is an electron harvesting machine. That's the way to remember it. All right, but now here's where it's going to start getting really interesting. That so far was all the buildup to understand why are we talking about this. Now here's where it gets really interesting. In some primitive bacteria, Krebs cycle goes backwards. So you have an anaerobic bacteria. It'll run Krebs cycle backwards and take those CO2s and be adding them in to make bigger carbon molecules. So anabolic is to build things up. Normally, like in humans, Krebs cycle is catabolic, to break things down. So anabolic metabolism enables the cell to run Krebs cycle backwards and to make bigger molecules, like it can make acetyl-CoAs and then use those for making fats, for example, and for making other things. So why does this matter? Well, a cell, like a bacteria, just wants to grow fast. It doesn't care about doing a job as a multicellular organism. It doesn't care about helping the liver. It doesn't care about helping anything. It's like cancer is like a selfish bacteria that's become more primitive, dedifferentiated to become like a selfish, primitive, anaerobic bacteria that only wants to grow for its own sake. Okay, so this is a key point, that Krebs cycle will run backwards with cancer. We're not going to get too much into reverse Krebs cycle today, but that's a good thing to know, that cancer is a reversion to a primitive phase of a cell, so it becomes like an anaerobic bacteria. That is very useful to know. That's essential to know for having a good sense of cancer. All right, oops, crap. Just want to show you a little bit about like how would I memorize Krebs cycle. We talked about acetyl-CoA coming in, the C2 molecule, meaning 2 carbons. It's going to combine with a C4 molecule, 4 carbons, called oxaloacetate, and it'll form citrate. Citrate has a very unique structure. There are 3 carboxylic acids on there. So it's called tri for 3 carboxylic acid. So 1, 2, 3 carboxylic acids. They're all deprotonated, so it's called citrate instead of being called citric acid. All right, I made these in blue to show this is where the acetyl-CoA carbons come in. All right, and the way I drew it, doesn't it look a lot like a triglyceride, like a glycerol molecule here, which is like a propane, 3-carbon alkyl group of organic molecules, and then these are like the fatty acids sticking off of it. Anyways, that's just a little bit interesting. So it's a tricarboxylic acid, and that's why it's also Krebs cycle is also called a tricarboxylic acid cycle. And because I can call this a tricarboxylic acid, is another name for it as well as citrate. Remember the T right here, and I'm going to use that to make a mnemonic. So the word tia means ant in Spanish. So this is my tia, okay? T-I-A. And so the T is for tricarboxylic acid, T for tricarboxylic. The I is for isocitrate. That's the next molecule in the Krebs cycle pathway. The A is for alpha, as in alpha ketoglutarate. And then the next 2 molecules are going to start with S. S. S for succinyl-CoA, then S for succinate. I just wrote starch solution on here like Dr. McDougall's book as just a quick reminder of the SS letters. All right. Then we're going to go to F for fumarate is down here. M for malate is over here. And then we're back to an O. And I'll use this as the O. So this is a simple pictorial mnemonic, almost an acronym. TIA. So here, imagine I'm talking to my aunt. Tia, starch solution, FM radio, it's on, okay? So if you remember that, you could just look at that and say tricarboxylic acid, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. Boom. You got all the things in the cycle there. And once you've got all the things in the cycle there, then it becomes easy to remember all the stuff. I like to make a mnemonic to remember a cycle, so then I got all the molecules in my head, and I can start to think about more details if I want to. For our purposes, we don't really need to get into too much of the detail, but just know that Krebs cycle normally runs clockwise, and it's harvesting electrons as its main purpose, and its secondary purpose, it's chopping carbons off into CO2, and then we exhale those out of our body. And this is the foundational step. All of human metabolism revolves in a circle around Krebs cycle. It's the center. If you look at a giant thing with all the reactions in human metabolism, Krebs cycle's right in the center of it. Okay, and these harvested electrons are now headed over to electron transport. So these are going to go to electron transport, and these are going to be pumped out as protons. Okay, here's another picture of electron transport. So basically, here's complex 1. Electrons are going to be passed down like a gradient here, then the coenzyme Q, then the complex 3, cytochrome C, and then on down to complex 4. At each of these stations, proton is pumped out. This proton's pumped out into the intramembranous space. Here's the outer mitochondrial membrane, OMM. I should have put an O there. And this is the inner mitochondrial membrane, IMM. All right. Then these protons are pumped into intramembranous space. They build up a very high pressure gradient. It's a chemical gradient because of the concentration of protons in the intramembranous space. And it's also an electrical gradient. And so these protons really want to get back into the matrix. And when they come through this protein right here, this is called ATP synthase. They come through with such force that it provides energy that can add this phosphate onto ADP to make ATP. And ATP is the energy currency of the cell. Again, it's like a $20 bill of cash. So this is how electron transport works. This is the electrons being transported, and then the harvesting of the gradient, and they call it oxidative phosphorylation. It's oxidative in the sense that oxygen is going to get the electrons as the ultimate acceptor of electrons. That's going to enable it to be converted to water. And then the harvesting of the gradient is the phosphorylation component, that you're adding a phosphate, phosphorylating this ADP to make ATP. And that's how life exists. That's how all multicellular life exists on this planet. And this is electron transport with oxidative phosphorylation. A lot of times I'll abbreviate it ETC for electron transport chain, and then OXPHOS for oxidative phosphorylation. And that is how ATP is made. And then I really like this diagram because it kind of shows you break those hydrogens off of the glucose molecule and then its subsequent derivatives, reaction products, and then you send these electrons to electron transport, and they run through progressively more aggressive grabbers of electrons until they get down to the most aggressive grabber of electrons is oxygen. And this is why cells absolutely have to have adequate oxygen to run this metabolism, to get energy to do all the jobs they need to do. And if they don't get this oxygen, they cannot function as a multicellular participant in an organ system inside of a human. And they're either going to die or they're going to transform themselves into cancer. And that's why one of the biggest things I've talked about since the very beginning is you need good tissue oxygenation. And that's why you want to be very low in fat, because you get more fat decreases oxygen delivery to tissues. You also want to be lower in sodium because it causes vasoconstriction, and you get less oxygen delivered to tissues if the sodium is quite excessive. And the body can compensate over mild amounts, but you progressively keep building up atherosclerosis and advanced lactation end products when you start getting diabetic physiology, and you start progressively worsening oxygen delivery to your tissues, and then you put yourself at risk for a Warburg effect, Otto-Warburg effect, of tissue hypoxia inducing cancer. And so in the reverse point of all this is by maintaining good blood flow to all your tissues, you heal all your problems better, and you're much less likely to get cancer, and you simultaneously drop your animal protein intake, then you drop all your growth hormone effects, your anabolic effects of animal protein, and you help prevent a cancer from growing. So that's the big picture here, but the real message of this talk is you harvest hydrogens from glucose, and then you take those hydrogens and you use them for both parts of your electron transport. To run your electron transport chain to generate energy that's going to then be given to O2, and then to pump these protons. Okay, so what we went through here was the key points on how human metabolism works. Electron transport chain leading to oxidative phosphorylation. Here's the oxidative component, here's the phosphorylation component. And that's how life on Earth exists. And cancer is a disaster. With cancer, you don't have enough oxygen going to these cells. Krebs cycle goes backwards, and bad things happen. So we're going to talk about that in subsequent videos, but I just wanted to establish how electron transport works and oxidative phosphorylation, and how Krebs cycle is related to that, and the fact that primitive life goes backwards and cancer goes backwards on Krebs cycle.